So welcome back to the final screencast for Chapter 1, looking at the scientific method. I'd like to go back to this chart and go a little further into this next arrow, and one that's not shown on here, about what happens next. So we had the Pfiesteria as a possible cause of the death of 1 billion fish. Data was collected, results were interpreted, and, excuse me, if the results did not show that Pfiesteria was a cause, then that would mean the hypothesis is rejected and we would go back and form a new hypothesis. So, for example, we might form a new hypothesis that says water temperature is the cause of these deaths. But even if the hypothesis is accepted, there should be another arrow in here where we still go back to observing and questioning. So let's say the results showed that Pfiesteria did cause the death of 1 billion fish. We might want to question, how did that happen? Did they produce a toxin which killed and dissolved the flesh of fish? Did they do something else? So that brings us back to another round of questioning, hypothesis formation, data collection, etc. So if we think, for example, maybe the Pfiesteria produced a toxin and that's what killed the fish, then we would form a testable hypothesis. A toxin killed the fish, a toxin produced by Pfiesteria. We would design an experiment to test that. Maybe we would take an aquarium, grow a bunch of Pfiesteria in it, then filter out the Pfiesteria and just take the aquarium water, which may contain a toxin, and introduce that into an aquarium with fish and see whether there was something in the water, like a toxin, that then killed the fish. So there we would be conducting another experiment to test our prediction and collecting data. And if the fish did die, then we would interpret the results as, it looks like something that was produced by the Pfiesteria did kill the fish. And then we would go on to either observe and re-question and possibly at the same time disseminate findings. Okay, so now that we have the scientific method as a baseline, we need to look at the types of experiments that can be performed either in a laboratory or out in the environment. And ideally, what we would have is an experiment that could be conducted in controlled conditions such as the laboratory. That's called a controlled experiment. It's controlled because what you do is you have everything be exactly the same except for your one variable that you're testing. In environmental science, sometimes we can't do that. The environment is a very complex system, and we may not be able to duplicate conditions in a laboratory. So that's where natural experiments come in. It's when a natural event, for example, a volcano, acts as an experimental treatment in an ecosystem. But ideally, we'd really want to come back to this idea of a controlled experiment. So next we'll look at an example of a controlled experiment, an example of a natural experiment, and discuss those a little further. So let's start with the left, which is a controlled experiment, and on the right you have a natural experiment. On the left, you have your question. Do organophosphate pesticides have a detrimental effect on central nervous system? Don't worry too much about null hypothesis. We're not really going to deal with that in our course. So we'll conduct an experiment where we have two groups of organisms, the control group of mice and the experimental group of mice. So the control group and the experimental group need to be matched as closely as possible. So let's say you have 5 females and 5 males in your control group. You need to have 5 females and 5 males in your experimental group. Let's say you have only adult mice, but not too old, a certain age range in your control group. You need to have the same age range in your experimental group. Let's say you give certain diets, certain food, certain amounts of water to the control group. You need to do the same with the experimental group. The only difference should be the experimental variable, which in this case is adding a specific amount of a pesticide, in this case, I'm not sure how we say that, chlorpyrifos, to the experimental group, and it's not given to the control group. Then, as the experiment is conducted, we measure the enzyme activity in order to test for the effect on the brain. If the results of enzyme activity, the data, indicate that enzyme activity is reduced in the experimental group, but normal in the control group, then we could interpret the results that under these conditions, feeding this pesticide, they did young rats here, to young rats reduces the activity of a key brain enzyme. So that means the experiment is, the hypothesis is supported. If you want to get into it, the null hypothesis is disproved. So that's a pretty good experiment because it gives us a really good idea. It doesn't tell us everything. For example, that's the effect on mice. Is it going to have the same effect on birds? Is it going to have the same effect on humans? All of these are further experiments that would need to be conducted, and we don't have information simply based on the experiment on the left. But it's pretty good. On the right, we have a natural experiment. So here we have Mount St. Helens before the volcano erupted. Then we had it as soon as the eruption occurred. This picture doesn't really show it, but really a huge part of the mountain actually just disappeared, just sloughed off. And here we have a regrowth of various organisms, particularly plants, over an area that had been completely covered with volcanic lava. So in this natural experiment, we might be able to determine what types of plants first regrow in an area that was wiped out, in particular in this case due to volcanic lava, but maybe also due to something else, like we might extrapolate it to see how plants respond to an area that was paved over. So that is a natural experiment, interesting but more limited in its ability to inform us. So in environmental science, we can sometimes do controlled experiments such as this, but other times we have to rely on natural experiments because we aren't able to recreate a particular situation from the natural world into an experimental laboratory. And in the natural world, the number of factors are going to be quite numerous that affect the results are going to be numerous. So let's take a look. So the unique challenges that are presented in environmental science. First of all, we don't have a control planet like we had that control group of mice. We don't have baseline data. There's no Earth 1 and Earth 2. There's not much data from how the natural world or the planet existed 200 years ago, 400 years ago, 1,000 years ago, 8,000 years ago. So the no control planet, no baseline data is a big challenge. And it gets complicated because it's often difficult to decide whether one thing is better or worse for an environment than something else. There are many, 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 many interacting parts to each question, which we will talk about as we go through the semester. Another issue is that it can be a challenge for economically developed and economically developing countries to perform or implement solutions. So developing countries may not have the money, but economically developed countries may have the money, but do they have the will to implement solutions? And last, many problems have to be solved not through science, but through political and social change. Science should inform the political and social change, and I hope that it will. But solutions are generally not just achieved through science. Definitely political and social change is required. So I'll wrap up this segment with 2 more slides. First, this is a picture from the edition 1 of Essentials of Environmental Science. And this is showing what happens to our electronic waste. In the United States, much of our electronic waste is shipped out to developing countries where it is dismantled, often in unsafe conditions using acids, heat, fire, and sometimes causing health effects to the individuals who are dismantling these electronics. Adding to this problem, some of the electronics are manufactured in a way that makes them very difficult to dismantle, and that leads to further problems. Third, many times because the electronic parts involve tiny parts, child labor is used to dismantle electronic devices. And last, what happens to all this waste once they collect the precious metals that are found in electronics to sell off? What happens to all of the rest of the waste, all of the plastics and other components? Well, often they are just buried in a dump, a hole in the ground next door to the community. And some of that pollution may work its way, and does work its way, in through the soil and into contaminating either the soil and or the groundwater that the people rely upon. So the overall message here is that it is a global community. We do need to think about our responsibilities every step of the way. Sometimes we don't have a choice. I tried to find out where my HP printer was going to be recycled and how, and that information was not available on the internet. So I did not have control over what happened in that instance. But where we can figure out how to implement solutions, I hope we would all agree that that's what we want to do. Okay, last slide. Key concepts. This is just showing you some of the things that we need to, look at. So you need to be able to define environmental science and what challenges we face. Define an ecological footprint and examine your own and those of your neighbors. And how do economically developed and developing countries compare and contrast? You need to be able to define the 5 key environmental indicators and how they are indicators. That means a sign like a thermometer, but maybe not tell us what actually is the cause. And discuss the concept of sustainability and its importance in being able to continue for future generations. So that's it for Chapter 1.